All right, I'd now like to introduce Dr. Thomas Durcan. He's assistant professor of the Department of Neurology and Neurosurgery, co-lead with Dr. Edward Fawn of the Early Drug Discovery Unit, and recent recipient of the Cyril and Dorothy Joel and Jill Reitman Foundation Prize for Open Science.
So when I was asked to do this talk, it was kind of like, how do I start? How do I pitch it? So in a way, I'm going to talk about two things. One, I'm going to explain how we get into this kind of early drug discovery and why stem cells is really at the core of it. And then I'll kind of introduce you to the different groups at the Neuro and also what we do. I know today we're kind of here to talk about partnering with the Douglas, about open science, but the truth is it's been kind of ongoing for many years. Me and one of your colleagues, Carl Ernst, have actually been working very closely for the last few years, and he was really one of the kind of seminal characters to actually help us. We've been doing a lot of exchanges of SOPs, assays. His team actually does some work in our lab, and then vice versa, we're helping him in some projects. So it's kind of already started in a way for that. But I kind of want to go back to something kind of Jason talked about earlier, and that was kind of the whole degenerative disorders versus cancer. So at the Neuro, and in particular our group, we really are focused on neurodegenerative diseases. Here it's obviously neuropsychiatric diseases, and these have a devastating effect on millions of people worldwide. At present, like pretty much what Jason's shown was true. You really have limited treatments and you don't actually really have any defined cure for these conditions. L-DOPA was discovered in the 50s for Parkinson's and it's really become the gold standard since for actually managing the symptoms for patients. And as I says, according to the World Health Organization, they do predict that cancer will surpass, sorry, by 2040, they do predict if there's no real new clinical interventions or new diseases or new treatments, that diseases of the brain will surpass cancer as a second leading cause of death in Canada by 2040. So why is this? When you actually compare the two diseases of cancer versus CNS disorders, there's actually three main differences between the two. When you look at cancer, it's really a disease of gain. Every patient that actually has a cancer tends to actually have more cells. From these cells, you can actually biopsy them, you can isolate them, and you can grow them in a dish. And from here, you can do a lot of cool science. You can deeply phenotype them, you can look at the protein, the RNA, you can actually develop assays on them, and you can really start to get to more personalized precision medicine. And that's really led to an explosion in new therapies for cancer over the last kind of 2 decades. When you look at neurodegeneration, though, when I look at Alzheimer's, when you look at Parkinson's, ALS, it's actually a disease of loss. The cells we want to study are actually lost within this disease. You actually have a decreased number of neurons. And that then actually causes a problem. So one is you don't have the cells that are affected, and B, they're actually in the most inaccessible part of your body within the human brain, surrounded by this hard case, like this big skull that to actually get in there, it's actually quite difficult. Like when you have a big head like mine, it's quite a hard nut to crack. And it's not like I can actually just go up to people and go, hey, I need 1,000,000 neurons. Can I get them for an experiment on Tuesday? People tend to be very attached to the neurons that they actually have. What they have tends to be what they actually need to survive and actually do their day-to-day life. And so that's actually led to problems in terms of access to material, and the material we do get is very limited. So Jason, as a pathologist, they do get clinical samples all the time. So we do get neurons, but it's very limited. And often it's actually from patients that already have diseases, because if you're getting an actual surgery on your brain, it tends to be you either have epilepsy, you either have other neurological or neurodegenerative disorders that require this type of clinical intervention. And so we kind of, when it's come for the last 3-4 decades, when I actually got into studying Parkinson's in 2007, we always wanted to work on human neurons, but because we couldn't actually get the neurons, we were really stuck with working on two different models. One was the immortalized cell lines, HeLa cells, HEC293T cells, U2OS cells, and these have been great. They've been around since the 60s and they've led to an explosion in the biology behind what these diseases are. But the problem is, you'll notice it's a cancer cell. It doesn't actually have any features of a neuron, really. Or we have our little furry guy on the right, the rodent, the mouse or the rat. This guy has won the Nobel Prize probably close to 20 times over the last century, and I'm not ever going to short sell his achievements or her achievements. But the issue is also that when you try to compare neurons from a mouse versus a human, they're actually very different. They actually do look alike, but the more we start to study the similarities and differences, we're actually seeing that they're actually quite different in terms of, let's say, neurotransmitters, genes expressed, or even just simply responses to drugs. And so we've always really wanted a human model for human disease. So let's say we work on blood disease, we always wanted to have a hematopoietic cell. For heart disease, you'd want a cardiomyocyte. For neurons, we want human neurons on a dish or in some other way that we could actually then start to study the human disease itself. And really, this is where stem cells come in. They actually start to make this possibility of creating human models for studying CNS disorders possible. And so what is a stem cell? Many of you probably already know what a stem cell is, so I'm just gonna go through this quickly, but there's two main features of what a stem cell is. One is it replicates itself. It divides and grows constantly. So you can actually, it's almost like an immortalized cell, but it kind of gives you almost an infinite amount of material when you need it. And it's pluripotent. This pluripotency is its key feature. It's like baking a cake. If you give it the right ingredients, you can actually direct it to where you would like it to go. You can make a blood cell from it, a pancreas cell, or in our case, you can actually make a neuron. And so this is where a lot of the work came about 10 years ago. It was actually seminal work from Shinji Yamanaka in Japan. He kind of knew from the work from John Gurdon in the 60s, you could reprogram a cell. You could take a skin cell and bring it back to almost state zero, get this pluripotent stem cell in dish, and then use it to actually then make any cell type you want. And he spent a few years actually taking mouse skin cells and then reprogramming. He threw 24 different transcription factors into it and found 4, which later he called the Yamanaka factors. A year later, work from both his group and James Thompson's group led to 2 seminal publications in both Cell and Science, where they were able to identify human iPSCs. And since then, we've only been since 2007, it's been nearly 12 years. We've seen an explosion in both the actual number of publications, the number of groups, and also the number of therapies that are emerging through human iPSCs. And this has led to the Nobel Prize in 2012 for John Gurdon and Shinji Yamanaka. And the big benefit is you can get 3 main cell types. You can get anything from the endoderm, the mesoderm, or the ectoderm. And they've really ushered in a new era in brain science and drug discovery. So back when this started, it was always people were getting skin biopsies. So a patient or a person could come in, you would get a block of skin, you would dissolve it or dissociate it, you would grow fibroblasts for a month, and then you could reprogram it then into this human iPSC. Yeah, into this human iPSC there. Later on, we actually decided, okay, people tend not to like getting like an actual piece of skin extracted. Blood draws are a little bit more amenable, although if you've ever sat for 20 minutes for a blood draw, it's never the most fun as you see the little red liquid come out of you, especially, that's my opinion. Plus, I've actually got deep veins, so if a phlebotomist ever sees me, I'm like their worst nightmare. But we do get blood at the Neuro, and it's been a kind of constant feature and actually a great benefit for us working with the CBIG team is because we can get blood, we can then reprogram it into iPSCs. And it's actually urine here. This is actually all stemming from the work of Carl Ernst. He actually years ago came to me and was like, Tom, if you piss into a cup, we can make stem cells. It was like, oh, sorry. And I was like, okay, Carl, that seems a little crude. And then he explained the process. He was like, no, from urine, you can get epithelial cells, you can grow them for 10 days, and then you can actually make them into iPSCs. And so this is really where we're going to, is with these iPSCs, you can then get 2 types of cells, either 2D human neurons or 3D neuronal organoids or neurospheres. And so this is really what we've been trying to build over the last 4 years. The first few years is kind of building the infrastructure, the team, all to really get towards building kind of target-driven precision patient models to identify lead therapies. And it all starts with the patient. Without the patient, we really wouldn't be doing any of this at all. And because we have the patients that are so willing to actually give their cells, we've been able to make quite a substantial collection of these patient-derived stem cells. From here, you get these patient-derived neurons, and these are actually just dopaminergic neurons that we actually use to model Parkinson's, which we then have been bringing into 2D small molecule screens, ultimately leading to a few compounds. And then the goal was always to, you take your lead compound, and then you would start to actually go into your mouse model. And then we were thinking about it, was like, why would we do all of this? Like, we've gone and done all this stuff of human model, human compounds, and then we go back into an animal model. So we actually started to actually think about how we could repivot, because obviously, a lot of times when we actually take the mouse, we then bring the drug in, then go into the patient. A lot of the time it's actually failed for both. At ALS, it's failed 18 out of 20 times. At Parkinson's, they've got a graveyard of 25 drugs that they've treated the Parkinson's mouse all the time. Then it goes into the patient and it fails. So we thought, if everyone else is doing one thing, why don't we try something different? And then this has kind of led us to actually build our, what we call our 3D kind of discovery pipeline, where we try to find stuff in 2D, validate it in 3D with organoids, and then ultimately try to get towards lead compounds. We're not saying the mouse or the animal models are obsolete. We're actually working quite closely with some animal researchers. So Gary Armstrong with zebrafish. We actually do have different models of mice as well, and they've also been adding in the marmoset models as well. So let's say we can really find 2 human-based models to validate a therapy. It starts to increase your confidence when you go into an animal model at a later date. So why do we want to work on organoids, or mini brains as people like to call them? So why 3D? And there are several reasons why we decided to go into this kind of structure. One was it's interconnected. We were doing 2D neurons for many years. They're flat. When you have a brain, it tends to be actually quite spherical, mushy. And so you kind of wanted this interconnectedness. We also wanted something that was quite scalable. When we were doing 2D neurons, we found it actually took a lot of effort to actually get multiple plates. When we just wanted to scale this up to get large amounts of tissue with an organoid, it was actually quite easy. We've now got 26 lines ongoing from different patients with about 8 to 10,000 mini brains growing at any one time. And then next year, we are developing the capacity to scale this up to anywhere between 20 to 30,000 based on projects and funding available. They're easy to maintain. Pretty much once it's growing, you just have to change the media. Once the media is changed, they grow as long as you want. We've taken our oldest was actually 18 months old, and the only reason we stopped it was because of pretty much curiosity. We just wanted to see what an 18-month mini brain would actually look like if we started a section and see how it would look. The oldest on record was actually Paola Orlotta at Harvard. She actually grew some for 4 years. So that would be a great graduate project, I guess. Start your organoid culture, leave for 4 years, come back, and then do your experiments. Although I'm not sure your advisor would be super happy with that. They're very diverse. When you grow flat neurons, they actually tend not to have, they're almost perfect. You get the perfect plate of 80% high quality dopaminergic neurons. But in reality, your brain is a kind of melange, a very mix of different cell types. And when we actually let these organoids to form, that's actually what happens. They try to self-organize and try to actually form a lot of the different cell types you want. And they can be customizable. So they're little pieces of tissue. But when you give it the right developmental cues, you can actually get it to form for different regions within the brain, either the forebrain cerebral, the midbrain, the hippocampal, cerebellar. It all really depends on what your interests are and what your needs would be. And so this is really where we've been going with our discovery research, and led to kind of 3 main core values for what we want to do. Is one is to really drive collaboration for research on these disorders. So yes, we have a large team now, but a lot of the stuff, the reason why we do it is because we go out and find PIs to actually work with us. And now PIs just come to us. They come up projects, and we try to essentially develop tools or assays or something that we can use with the stem cells to really then go towards the next step, which is actually to build the next set of innovative tools and models. Because when you look at the likes of Harvard, MIT, the reason why they're successful is because they have people building the new tools, and they test them. Then from these tools, they get the big paper, and then they make it available, and then we get it. And by that stage, Harvard's already moved on to the next thing and the next thing. So our goal is, how do we actually catch up at these big institutes? It's not to try replicate what they're doing, it's to try do something completely different. Try take almost risks and try find the latest and new idea that's no one working on, and actually try and see, can we develop it either in a dish, which has led us to actually do quite a few new things. One is to work with the biomedical engineering department quite closely. So we're making a lot of new devices, fluidics, even adding electrodes or other stuff with the cells, or to actually go work with the computer people. We're in Montreal, so we have huge groups of people with AI experience, with informatic experience. These cells, these human neurons, these organoids, they're a huge resource of data, but we haven't even touched it. Like, it's like a gold mine of data that we haven't even scratched the surface of. And that's something that we have to do a lot better of, to actually start to model and understand these diseases in a dish. Because ultimately, the goal is we want to start to translate these models into new therapies for CNS disorders. And so we've really, we started as the MNI IPSC CRISPR platform, and over the last few months, as we've kind of grown, we've started to kind of change our name to NeuroEDDU, Early Drug Discovery Unit, to start to really focus on what the kind of mission was, because we were working in a lot of different areas, and we wanted a kind of catch-all name to reflect everything we were doing. And this really fits with the whole idea. We're actually, at the core of it is stem cells, or induced pluripotent stem cells, but we've really broken down to an almost miniature academic pharma system, where we have 4 main working groups, all interconnected, all working very closely with each other, but all working in kind of slightly distinct areas. One is either to phenotype these iPSCs. So once we get the patient cells from CBIG, once the stem cells are made, they have to be deeply phenotyped, characterized, banked and then put into CBIG for dissemination to the wider public, and also CRISPR editing. There's many repositories worldwide. You can get stem cells. I'm not saying you can't, but oftentimes when you ask for the isogenic control, it's missing. If you want an actual rare disease mutation, it's not made. And so we've really kind of over the last year and a half, took the time to actually build the staffing with the guy Eric Danon from Toronto to really actually build this. And in the last year, he now has the pipeline in place where we've actually built and edited nearly 35 to 40 different IPSCs. And that's now really led us to scale up the work in terms of making these stem cells and also putting them into CBIG. As an early drug discovery unit, also a big part of it is discovery assays and screens, and I'll talk about that in a minute as well. Organoids and tissue engineering is another group, and training and outreach, which I'll talk a little bit about at the end. And so everything really where it begins is here with the patient. When the patient comes in, they give blood. The blood then goes to here, these vials. These are the ones that go into CBIG, and this is where we start to come in, where we take the blood from them. And you can actually, if you ever want stem cells from CBIG as well, you can easily put in requests through Jason and his team. And from here, you can actually, once you want it for stem cells, you get them, you make them, and the stem cell is what you then use to actually do your work. So we started this probably about 2016, and over the last few years now, this is pretty much just a quick summation of the catalog. I actually never had this until last week. It happened that I was giving a talk elsewhere, and then I started to actually put together all the different lines we'd actually make, and I was like, wow, we actually, over the last 3-4 years, have made nearly over 82 IPS lines, either from patients, through patients, and then correcting the lines, or simply through CRISPR editing. So on the, just from healthy controls, we've got over 18 cell lines. From the Parkinson's side, it's close to 36 cell lines, where we've covered many of the actually high priority mutations that people are interested in within the field, either with LRRK2, GBA, PARK, and PINK1. We even have sporadic lines. And we've even gone and knocked out a lot of different genes, because a lot of people, the interest is in a lot of these diseases, if I take out the gene and it's a loss of function, what happens now to the neurons and the cells? And on the right here is just a lot of the different genes that are actually involved. These are rare mutations we don't have in our patient population. So we've actually made them and introduced into 2 or 3 of our main control lines that we use as our kind of master lines. And we've done this the same on the ALS side. We actually have SOD1, C9orf72, FUS, VAPD. We have a lot of different knockouts. We have a lot of different knock-ins as well. The one line that I actually want to point out, it's the proudest achievement. I only found out about it last week, was C9orf72, because point mutations are actually quite easy to fix. C9orf72 is an expansion mutation. It's really long. It's in the 5' UTR, I think, or maybe it's in the early 5' end of it, but it's a long region. But Eric actually and his team, they actually took it and they actually knocked it out. They actually took out the piece to actually make it into an isogenic control. So now we actually have the exact isogenic matched with the disease line, and only 2 or 3 groups anywhere in the world actually has this. And so this will be made available for groups to use then next year. We've also kind of added to the collection, not just through internal lines, we've also added a lot of external lines as well. We have about 60 cell lines. We then, because we've been building our collection, we've learned the mistakes that other repositories do that we actually then try to avoid ourselves. And we can keep that for the questions if you ever have questions about what repositories do that we don't do. And then a lot of this is also whole genome sequencing is now underway. So we have about 30 of these lines that are now undergoing whole genome sequencing, both at the stem cell level and the PBMC level. So we actually can understand what's going on at the whole genome level itself. But we're not just kind of numbers of stem cells are great. So, we can get 100, 200 IPSCs, but it's no good if they're crap. And the reason why we really kind of wanted to focus on actually making sure we had good IPSCs was that led us to build a quality control pathway and focused on 4 main areas. Cell growth. Every cell that we make, they tend to grow differently, and that has to be reflected in how you study them and also in the kind of initial considerations. Some cells grow better on one media than another media, and so that has to be accounted when you're working with them. Pluripotency. Oftentimes people say, I've got a stem cell, it's great. And then you go, is it pluripotent? And they go, oh, well, they put on the antibodies. And then you look at it and you're like, yeah, it actually isn't really that pluripotent. So you probably shouldn't work with this. That cell here is actually a lot more pluripotent and should be a better cell for you to work with. And the most common defect or problem we've heard from many researchers, I actually did a stem cell training workshop last week, genome stability. We had a student, she did a year in a project. with this cell line, and she thought it was all great. And then she did karyotyping and realized she actually had an entire extra chromosome, because with these cells, chromosome instability creeps in all the time. When you reprogram a cell line, it resets the genome. And so you have to take really close attention to not just like overt, massive karyotyping or chromosomal abnormalities. Sometimes you even just get extra duplications of certain genes. And so there are tests out there that you can actually screen your cells on a constant level to make sure the cells you work with are all the highest quality as possible. And in reality, the last thing is the main feature of why you need it, kind of differentiate. So there are trilineage testing, we do neuronal testing, you can do both image-based and qPCR-based profiling to show that your cell, it's an iPSC, so you want a cell type. If it can't make the cell type you want, then you're probably going to have problems then later on. And so once we have these, so that's the idea. You can have your stem cells, and then you want to make it into 2D neurons. And so this is really what we focus on. We actually have focused around 3 main disease areas: Parkinson's, ALS, and developmental disorders. And the real reason we did this was these were the patient populations we had greatest access to. For studying Parkinson's disease, we focused on making the 2D dopaminergic neurons. For ALS, we focused on making the motor neurons. So we actually make them for Axel, a company out in the UK, and they actually distribute them as well. So we've actually built the SOPs for this too. In terms of other cell types for ALS, we also make astrocytes and microglia, which we've only started to do recently on the microglia side. And then for developmental disorders, we do make cortical neurons quite routinely. And something we've actually started to do in collaboration with Geneviève Bernard and others at the MUHC is oligodendrocytes. So we actually have staff members embedded within the gland site to actually make the oligodendrocytes for us. So we're actually doing that as a joint collaboration with them. And so a lot of this stuff, it's great. You can do it manually and you can do it 1 or 2 at a time. But the truth is, to actually do this consistently and to do this to scale, you need robots. I'm not saying that the students and the staff aren't talented, they are, but you always kind of need, actually, do we have... Okay. There was little videos here, so you see the pictures here. Well, yeah. Can I put it? Okay. Never mind. Anyway, take my word for it. We have robots and they move very fast in different directions. They put media in, they take media out, and they let us actually both code our dishes. They actually let us to do multiple wells and multiple assays in the process of 10, 15 minutes. Whereas if it was done manually, it would be pretty much 3-4 hours. And you would, by the end of a year of work of doing this, probably have massive carpal tunnel syndrome. It also lets us to manipulate, to process, to do actually our pitting of compounds, to do our immunofluorescent assays. And this is kind of the workhorse of it. So this is actually the schematic of what it looks like. The CX7, it's linked in with a robot arm, and we have a cytomate. So we can do both fixed assays and live assays. So we can do anywhere up to probably about 6,000 to 8,000 compounds at any one time if we wanted to. You can do, if you do a fixed assay, we can just put all the plates on the hotel. We can go home on a Friday and come in on a Monday. And the robot arm will just take the plate, bring it to the high content reader. The high content reader will read every well, and then we'll have all the data then from that. And then the data part is really the part that's been the big crux of it. So when you do all these screens, you get lots of data. And so this is partly what we're actually working with Dylan, Claire Brown, and others. So Jason talked about neuroinformatics. When it comes to fluorescent imaging data and all of this data, it's like the wild, wild west out there. Everyone has their ways, you know. I image my cells, I record the images and the data in a different way. We've been working with Claire Brown at the Advanced Imaging and Advanced Biological Imaging Facility, and also the Brain Imaging Center to implement Neuro actually within the MNI itself, and then to scale it up to McGill. I think it's something you also have here at the Douglas, and you're actually an early adopter of it as well. And that's something we'd also be kind of interested to kind of get your take on how we can actually bring in Omera within our sites to actually start to really implement the data acquisition, data storage, and metadata, and see if there's kind of links. Because we're also partnering with Steve Ogg at Alberta, who's going to help us kind of guide through this process as well. Because we think it's going to be, over the next decade, as you get reams and reams and terabytes of all this data, it's going to become more and more critical to actually have the ability to both store, curate, and disseminate this type of microscopy data. Because of the automation, we've been able to do some screens. This is just an example of one screen. It was actually, I always like to show this because it was the first screen we did. It was actually a synuclein uptake assay. So with Parkinson's, you have these Lewy bodies. It's a feature of the disease itself, and Lewy bodies are little aggregates that form it in the brains of patients. You can make kind of artificial aggregates within a dish itself. You can label it with a fluorescence tag, and you can kind of just see is it actually getting into the neuron itself? Because the idea within Parkinson's is these synuclein aggregates can get passed from neuron to neuron. So we then wanted to block the uptake into the neuron itself. So we actually screened what we call a low-pack screen. It's actually the library of pharmacologically active compounds. We screened about 1,300 in one of our first automated assays. It led to about 52 hits that have come from it. That was last year. And now, since then, we've actually partnered with a small AI company, in Vivo AI, to take these hits and actually find new hits. And gradually, through their AI algorithms, have been able to find new hits free of IP that we're now going to do lead optimization and see, can we start to actually, instead of doing large scale 100,000 compounds, can you start on more small scale compound sets and then scale it up to find newer and newer compounds bit by bit. On the ALS side, this is just an example of one assay we've been working on, is just a neuromuscular junction assay where you could take neurons, motor neurons in one side, you can take muscle in another, you can then connect it. So we've also then gone out and partnered with a microfab company, Nuvio. So these are the devices that they sell, and we actually co-developed this device with them. So when we say co-developed, is they wanted to make a device, so we actually agreed to then help them test it. And then from here, it's then led to the ability to actually culture motor neurons with muscle in these dishes. And from here, then we're actually starting to work with them to create more of a multi-well system, what we call the 96-well omega, that we can then actually bring into screening. And that's actually going to be part of a larger scale screen that we're going to be doing in 2020. And so the other part of this is the 2D side, but then you also want to do 3D. So we've really been focused on matching the 2D and the 3D. And in terms of Parkinson's, that meant forming midbrain organoids where they'd be enriched with dopaminergic neurons. In terms of ALS, we've actually been developing more of a spheroid model where you could actually enrich with motor neurons and other types of cells. And then on the actual developmental side, cerebral organoids. And this is how you make it. It's all really quite straightforward. If you ever work with IPSCs and can do 2D culture, you just separate them, you dissociate them into a single cell, and you form little aggregates in a low detachment dish. From here, then, it's just you embed it with some jello, you put it into a shaking flask, and then you just leave it for as long as you would need. And all you need is two factors, chemical and physical, and then that leads to your midbrain neurons. And this is just the process as you would go through it. And you can actually, from here, you can then provide nutrients and oxygen. So when we actually do it, we actually do a lot of shaking, and I'll see. Yeah, okay. So this is like the bioreactor we grow it in. So imagine it's little white balls and they shake around. It's like a kind of Christmas tree lights when you see snow whirling with wind. So that's my best artistic way of describing it, I guess. But the main thing is the reason why we shake these, so we can grow in one of these bioreactors, you can grow anywhere between 8 to 900 at any one time. They're under constant shaking because we want to always enrich it for nutrient availability and oxygenation. Because if you ever leave it static for too long, oftentimes you get cells dying off within the center, and that then becomes a bit of a problem. This is what they would look like over time. When you start to actually form the organoid at day 1, it's about 500 microns in size. And after about 50 days, it grows to full size, 4 millimeters. And you can do many different features of it. One thing I'll say, it never really goes above or beyond 4 millimeters, likely due to the fact that it doesn't have vasculature. And that's something we're working with some other groups to do. But you can do a lot of cool things. So you can section it. So we can section tissue, we can give it to the pathologist, and then that excites the likes of Jason and others because they're kind of always, they see human brains all the time, and now they can see these human pieces of tissue and go, huh, you actually start to see a lot of features of a human disease in this kind of tissue itself. You can also do cryosectioning where you can actually see neurons interdispersed. So the neurons are actually stained for MAP2, and we also stained it with red for GFAP for astrocytes. And this is actually a very young midbrain organoid here, about 20 days. They are active. So people always say, you know, neurons, they have activity. Do yours? And yes, they do. So if you actually take just, this is an Axion MEA system. It's a system that came out of Stanford and has these little copper grids. If you actually place your organoids on them, it can actually record field activity from your neurons. And I'm not sure if it'll play, maybe it will. No. But anyway, what we had 24 different organoids here. and you could just place them on top of these grids. And in these wells here, you'll start to see little kind of light blue patches or something. When you actually see it in real time, you could actually start to see the firing go on and off within the neurons itself. And so if anyone wants to see it afterwards, I'll be happy to show you. But what it means is that we're forming midbrain organoids. They have activity. They do have myelination if you let it grow to about 80 to 100 days. And then we're starting to explore, like, you know, how do they form tight synapses? What might a synapse look like within these structures? That's information we don't fully have just yet. And so really the goal is to build patient profiles over the next decade. We have a lot of time. It took many years to get to here, and now the real goal over the next decade is to start to build patient profiles on these mini brains, because when we think about Parkinson's, Alzheimer's, ALS, they're all rare diseases. In reality, there are probably 40 diseases and one for Parkinson's. For Alzheimer's, it's probably similar numbers as well, because each disease probably has very different subtypes. And so what the goal is to really get towards is to see is from their cells, what do they look like? First, to see, can we understand the disease from the cells and the structures that were being made, so the actual 3D mini brains themselves, and to actually understand the cellular jigsaw. And this is where single cell biology comes in. We spend all this time making it. Now we're actually going to spend a lot more time breaking it apart into each individual cell. Because by actually looking at each individual cell, you can actually start to understand what is it. What is it expressing? And then start to build more of a cellular profile on each of the organoids at each particular time point, to start to understand about developmental disease processes, maybe just even degenerative disease processes, and then start to compare these multidimensional profiles from patient to patient. And the real goal then is to get the really clinical trials on a dish, to actually say, if I found a lead compound and then I treat it in, because at the moment, the way people tend to do these type of studies is you find a lead compound and then you treat it with patient cells, or you take it and you bring it into a clinical trial and you see 5 patients respond, 95 don't, and that's seen as a failure. But maybe we're actually targeting just this catch-all is not really working. What we need to do is more of a stratified approach. Take your compound and start to see is which compound and which patient actually have the best response. And then from here, start to really build more patient stratification at the cellular level. And then with CBIG, the information they collect both at the clinical level, genetic level, even their drug treatments like profiles, we can actually build all of that in, start to build more personalized patient profiles combining clinical and cell data to then actually start to build more of a clinical trial on a dish. And so just the last part, so Dylan touched on it as well. So we have been over the last year working on making our work open and available. And so there was a question earlier about duplication and trying, you know, to actually avoid kind of replicating stuff. And a lot of this is really more, let's say you want to culture iPSCs in your lab. We don't really want you to replicate doing the whole stuff from beginning to end. What we'd actually like to do is take the guesswork out, make the actual protocols available. So if you wanted to work with iPSCs, you could take our protocol, you could try it in your lab, and then we could actually then work together and actually help define a way that actually problems that you might have developing in it yourself, we can actually get you to a much faster way to have it up and running within your lab, both in terms of the stem cells. We've also made a lot of our protocols for making the neurons available online, even how we make the synuclein fibrils we've made available online, so that people see how we do it, so they don't have to do the same thing over again. They can see what we do, they can actually still do it for themselves, replicate it, and if they have problems, we can then have ongoing discussions. And I think that's really what we're trying to get to, is not to not duplicate stuff, but if we're duplicating it, let's all have more active conversations with each other. Because if you're doing it, Carl's doing it, and I'm doing it, maybe we can actually have a kind of multi-conversation to see, well, if you're doing it and you're getting this, why am I not getting this? And then this helps really accelerate the work then from this. So far we've put 12 of our industry standard methods online. We've had about 2,800 method views, 1,200 methods downloads. We have a lot more to come. We've also put some software online. We actually do an in-house training program with myself and Carl Ernst, sponsored by Stem Cell Technologies, where we actually every year, 2 times a year, we actually bring in 10 people from around Montreal to actually learn about everything from iPSCs and about a lecture-based and a stem cell-based course. So far, we started in 2016. We started with 3 people. We're now about 35 people. We've trained over 80 plus users. This is probably well under the actual number. I always try to be more conservative. We have about 45 active collaborators going on at the moment. We have 8 industry partners, and we have 1 or 2 more to kind of come on board in January. And this is actually some of the different groups. We've really been able to leverage philanthropy to bring in academic and federal and provincial funding. And then also this has led to, because we've been able to get these grants, we can try high risk and high reward projects, leading to then industry coming on board to say, oh, you've got this cool thing, we want to actually work with you. And then how that's led to different companies actually coming on board to fund different types of projects. This is the team here. So Ted Fawn is actually the scientific director, also the director of it. I kind of oversee the day-to-day side. And this is the entire team here, covering everything from basic IPSC culture to CRISPR editing to tissue engineering, all the way up to even predictive modeling of the cells. And so we have a lot more online. So this is the website here. If you have a phone and you can click on QR code, you can then see all the data. We actually started putting up all our posters online as well. So if people go to conferences, the day after, we'll actually put the poster online. It was actually quite interesting when we were at SFN, people would come to me and go, oh, can I take a picture of your poster? I was like, well, there's no need. You can actually just go on our website, or if you see this code here, click on it, the poster's there. It's freely available. If we're going to put it out in the public domain, you can access it, you can read it, but all we ask is, please come back and talk to us. It's the whole idea of it's meant to be a two-way conversation, and that's kind of where we'd like to kind of maybe leave it there.